Hi, all. Welcome back. It's just one chapter this week, and it's very short. I think that there's a lot of things that we're getting into where they're hypothesizing about how things work, and I wanna focus on how we know things work. But then I'll show you some of the stuff we hypothesize. But a little bit about how eukaryotes regulate. We've done how bacteria regulate genes. Now we got to talk about eukaryotes. They don't have operons. So I want you to go back and make sure that you are comfortable with DNA organization in chromosomes. What is a nucleosome? What is a histone? What does it mean when we say condense the DNA? And then also quick review transcription and translation in eukaryotes. That way you have like a refresher in your head. And I feel like when we reviewed it the last time in lab that there was some not everybody was confident. Well, nobody was confident with what we were talking about. So I think this lecture will be very confusing if that doesn't make sense. Because I want to talk about how this structure, a lot of this lecture, is how this structural conformation of DNA affects the accessibility of some genes and not others. Because eukaryotes have a lot more regulation and a lot more times to regulate and ways to regulate. We're going to look at gene expression through several different stages because it's not just at the operon level. We don't have operons, right? We're going to have regulation at every stage until we actually finish the protein. We're going to compare and contrast bacteria and eukaryotes really briefly, look a little bit about the structure of chromatin and the nucleosomes and the histones. I want to start talking about epigenetics, which is our next week after you have the exam. That's our next set of what we actually need to talk about. The controls are outside of the chromosome. So we'll hint at that here. I'm going to give you a very brief overview of a model in yeast that we use for eukaryotic regulation. And then I want to talk about a database, and you can check it out and see some of the data that we actually use to understand human transcription, translation, and expression of disease as well. You might want to also, as you're thinking about what your paper is going to be about or your presentation, this may help. So I want to talk a bit about how we organize the process of regulating the expression of genes in a eukaryotic cell and how that compares to a bacteria. Remember, bacteria are single-celled organisms. They have a single chromosome, and it's in a nucleoid. So it's a ring of DNA, and we've looked at it with the PGLO system again and again. They have an origin, and then they have some places where we can insert genes in the ones we have been talking about. They have some different kinds of genes that they are expressing, and when they do that, that means that this is everything that this whole cell needs for its entire life. And this cell, when it's exposed to changes in the environment, can turn off a gene for this whole cell or on a gene for this whole cell. If you are eukaryotic and multicellular, which you are, you have many cells, and those cells all do different things. The cells in your liver do liver stuff, the cells in your brain do brain stuff, the cells in your skin do skin stuff, and that means that despite whatever is happening in the environment, those cells still need to make things and do functions that are important for those types of cells. Every single cell in your body has a full copy of your genome if it has a nucleus. And that means that every single cell has access to every single possible protein that you can make. But you don't want that. That's a waste of energy. You don't need liver cells making brain proteins. So not only do you need to have different things happen at different times, you need to regulate what part of your genetic library, your genome, each cell has actual access to, so it's not making things for the wrong cell. You've got to respond to the environment, but you have to respond to the environment differently depending on what kind of cell you are. And your DNA, because there's so much of it and so many chromosomes, and it's packaged around histones, and it's done all wrapped up differently, that's a challenge for your body to overcome or to use in order to access the proteins you need. If you do these incorrectly, this is a pathway to disease. You might have the right code for your DNA, but if you can't access it and use it appropriately, there's something wrong. So it's obviously the eukaryotic gene regulation is considerably more complex than in prokaryotes. There's a lot more DNA, there's a lot more times and places where it has to be regulated. Not only that, once you made a transcript, your transcript does not reflect the ultimate product of what your mRNA is going to be. You make a pre-mRNA transcript, you've got to put a 5' cap on it and a 3 polyadenate. That's the at the end of the tail in order to get it out of the nucleus. And then you have a lot of chromosomes that you have information on. You have a nuclear envelope that surrounds that information. And so there's a challenge of getting the mRNA from the inside of the nucleus, the nucleoplasm, outside into the cytoplasm. Here, at every stage in the eukaryotic transcription process, there are more things that have to be managed because there's containers and complexity that bacteria don't have. Eukaryotic genes are on chromosomes that are linear and stored in a nucleus. They are made up of DNA and histone proteins that the DNA is wrapped around. We got a picture of it here. And then non-histone proteins, other proteins that are associated with the DNA. And that complex of proteins and DNA is condensed into a chromatin structure. Chromatin is sort of the accessible part, and we've talked about euchromatin and heterochromatin, which regulates the different accessibilities. Euchromatin is truly able to be used right now. It's loose, it's out there, it can be accessed by RNA polymerase. Heterochromatin is all wound up really tight. You can't tell one gene from the other. They all look mushed together, and that heterochromatin is not accessible to RNA polymerase. So I'm gonna expand on what we talked about earlier, and I think at this point we're beyond what you learned in Bio 1. What we do know now is that we sort of used to imagine that the chromosomes were inside the nucleus in like a blob, and even when we did mitosis and meiosis way back in the beginning of class, we kind of drew during G1 phase, we drew the DNA as a little blobby place. That's not true. But we weren't there yet. During interphase and G1 of interphase, each chromosome actually has a specific location in the nucleoplasm, or the goopy part inside the nucleus where it actually hangs out. It's a discrete area. That means it doesn't have a membrane around it, but it's confined away from other chromosomes, and they don't mix and match. So it's not as chaotic as what we originally thought. And part of the reason that that's discussed in your textbook like that, especially for Bio 1, is until very recently we thought it was just goopy stuff. But now when we look, we can see that each individual chromosome occupies its own location, and then there's space between them, these interchromosomal compartments. This is areas with no DNA or extremely small amount of DNA, and this allows each chromosome to be in its unique space. Those structures of the chromosomes themselves are continuously rearranged. They're wrapped and unwrapped. The genes that need to be transcribed are revealed. That's the transcriptionally active genes. And then if they need to do transcription, the genes that need to be transcribed are open up, so they're revealed on the chromosome and not bound up in a lot of protein or functional group structures. And they're pushed to the edge of the cell's nucleus, so they're more accessible to the mechanisms for transcription. If there is something that you are never going to use in this cell, this is a liver cell, and you don't want to use any brain proteins, those chromosomes can be packed up somewhere else and wrapped up tight so they never accidentally bump into RNA polymerase, is my understanding of this. And that way that they're not just off, they're not in the right location to be transcribed. And this was new to me as of a couple years ago, so I imagine that this is new in the mid-2000s. There are little factories. So in the nucleus, on those edges where we're moving the parts of the chromosome that you actually need to transcribe, there's little factories of transcription where most of the proteins and polymerases that are regulating this process actually sit. These form and reform around areas that where the promoter, the starting region, actually is. And if I'm going to stop the process of transcription and I no longer need that transcript, I can disassemble in my cells these transcription factories really, really quickly. So I can stop the process even starting to transcribe without waiting for an external switch to trigger it. When I wind up my DNA, and we can say you too, we all do this together. And if you are not sure, if this doesn't make sense, this is actually something we discussed in chapter 12 for the, I guess, exam 3 stuff where we talked about, or maybe even exam 2 stuff where we talked about the structure of DNA and how it's organized. The DNA is wrapped around histone proteins. Those histone proteins are found in groups of 8. So they're an octamer. I have it real tiny, but not. Groups of 8. And they're, so I always kind of imagine them, they're drawing them kind of like puzzle pieces here, but like little pieces that you can wind your DNA around and keep them organized. I always imagine them more like a cat's cradle. So if you've ever played that game with your fingers, so you can wind the DNA around it. Depending on the shape and what is attached to those histones, we can modify them to increase transcription or decrease transcription, or to totally prevent transcription. So we are providing either more access to the promoter region where RNA polymerase can attach, or less accessibility to the promoter region. Histones are the proteins that the DNA is wound around. We can, hold on, give me some more words here. The histones have the DNA wound around them, and then the outside sticking out of them, they had their little tails of protein that stuck out. The N-terminus of the little proteins that make up the histones, so the polypeptide chains, are sticking out. And what that means is on the amino acid that is at the end of the structure of a histone, this end with the amine group, or the nitrogen, is actually sticking out, and we'll be able to either attach or one thing or the other to here to facilitate or inhibit transcription. If I add an acetyl group to this end side of a histone, it actually causes the positive charge of the molecule to decrease. That lowers the affinity of the histone to DNA, which is what charge? It's negative. So the DNA is negative, the histone is positive. But if I made the histone less positive, it holds on to the DNA less attracted to it, and that actually increases transcription. So I think I said this before when we talked about DNA structure. I just wanted to go into a little bit more detail because we have a little bit more information now. I'm changing the charge of this little group sticking out, and when I do that, it causes it to push away from the DNA a little bit, and that allows access of RNA polymerase easier to the promoter region. The acetylation of the nucleosome, it's actually catalyzed by HATs. Put a little acetyl group on the amine N-terminus of the histone protein, and that facilitates transcription. I feel like there's too many acronyms. Go with histone acetyltransferase enzymes, because that one actually says what it does. And I don't know if you're going to remember that it wears a fedora. Is it a fedora? We can remodel the chromatin too. This is moving nucleosomes back and forth. So these nucleosomes are balls of histones with the DNA wrapped around them. I can make them smush together or slide apart. So when and remember, all of this is about physical access of the RNA polymerase to the promoter. When I move my nucleosomes apart, this area is revealed. This can be with an enzyme, a remodeler enzyme, and some energy. I'm providing access to RNA polymerase. That's an enzyme that builds RNA, as well as, and we'll look at them in a minute, things that activate transcription in eukaryotes. They're activators that facilitate transcription happening faster. These sliding remodeling complexes, this SWI and SNF, are sliders. These sliding complexes loosen the connection between the histone and the DNA. They allow a bit of the gene to be revealed, and it reorganizes how the nucleosomes are attached to each other. The ultimate goal here, whether we're sliding or reorganizing the nucleosomes and variations of histones, is we are revealing the gene and the promoter of the gene that we actually want to transcribe. And so here I am not sure if this varies by cell or what, or if this is, some of these are human models and some of them are not. I know that I have always understood these models to be all options, but if I think about it, I feel like per cell, only one or two, or per gene, only one of these would be available, and I'm actually not sure. I'll look it up and see if I can find an answer. I'm asking myself my own questions here. We can also alter the structure of the DNA itself, not the proteins around it. So when I take my DNA and I methylate it, this means add a methyl group. This is a CH3 group. When I methylate something, as an aside, for those of you who are enjoying orgo this semester, methyl is not hydrophilic. It's actually hydrophobic. It is not polar. So what we're doing here is we're adding a non-polar functional group to this part of, we're going to add it to cytosine in this case. And what that does is, since it's not polar, it pushes away polar molecules like RNA polymerase a little bit. And it also changes how DNA winds around itself. So it actually, adding methyl to my cytosine, I methylate it to 5-methylcytosine, which happens where I have a lot of CGs, so a CG doublet, where there's two together. When this happens, this causes it to fold differently, the DNA structure to fold differently, and it basically prevents the structure of that part of the gene from being accessible at all. This is one of the processes we use during X inactivation, where we methylate large parts of the bar-bodied X chromosome. And when we do that, that makes it permanently inaccessible for the life of that cell. And we also methylate, and we'll talk about it during epigenetics, we also methylate some genes during the life processes of an organism. If I want to turn it off for the rest of your life, you're not going to need it, but you just needed it a minute ago. Now I want it off. Methylation may be an option for me where I can shut it down, and now you have no longer need or ability to use that particular gene. Cancer cells often, they're doing mitosis wrong. When they do that, that's because they have abnormal patterns of chromosome modification. They're accessing things inappropriately, they're dividing inappropriately, or they're making poison. I want to talk about the BRCA1 gene. The MLH1 gene is also a gene that it acts as a tumor suppressor or an oncogene. When they are having their normal function, and the BRCA, it's this BRCA, the breast cancer 1 gene is on chromosome 17 in humans. When these genes are doing without methylation, they're doing their normal function, they produce this protein whose job it is to move around the body and suppress the production of tumors. So their job is to make sure if you have a tumor, that tumor gets shut down early and doesn't get to the point where it's cancerous or metastatic, where it moves from one tissue to another. When these genes get inappropriately methylated, particularly on their promoters, that means that function is no longer activated. These are tumor suppressors, but during the process of excessive methylation, they become oncogenes. onco is cancer, genes is genes. They become oncogenes, and now their absence allows cancer to proliferate. Cis-acting sequences, cis means it's on the same chromosome as the thing it's controlling. So cis means it's on the same size. They regulate DNA expression on a gene that is not them, but a gene on the same chromosome. So as an aside, transacting factors are small RNA molecules that are being synthesized from somewhere else and regulating the gene. They're not exactly opposite. Trans-acting factors are crossing over from someplace else. Cis-acting DNA elements are coming from the chromosome itself. These are required, the cis-acting elements are required for regulating transcription of genes at the promoter. They also are involved in enhancers, which increase the transcription of the genes, and silencers, whose job it is to stop expression of the genes. Now, if you compare this back to bacteria, bacteria either were on or off. But enhancing is a process that we can use in eukaryotes where we make it more without turning it on and off. It's getting more processing. We know what promoters are. Promoters are exactly the same. Promoters are where your body knows to start making this protein. It's the start spot for DNA transcription. It is a piece of DNA, and this is really easy to forget because when we build the process of transcription, we spend so much time talking about the transcript. But remember, everything I'm giving you, kind of like with the lac operon, the basic structure here is going to be on the DNA. The promoters may be something like a Tata box, where there's lots of T's and A's. There's a core promoter, where we actually are grabbing on to an accurate start spot so that we can make sure that we transcribe the whole thing. There are nearby or proximal elements that modulate, so what it means, modulate the efficiency of transcription, make sure that I get all the way through and how many transcripts I am going to make. There are promoters that are focused. These are promoters that are right before the upstream of the gene that we want to do. They make one transcript and the gene follows the focused promoter. And then there are dispersed promoters. So there are multiple promoters in this example that if we start here, the transcript is going to be much longer than if I start here. And I don't have a huge functionality for this, except there's some variation in the introns and exons of these genes. The promoters are where we start transcription. If I look at this entire region, I've got a lot more going on than I did inside of my prokaryotic structure. And this is part of why this chapter is so short, because this is full of a lot of new information that we haven't talked about. And I'm asking you to kind of rewrite some of these processes in your brain. There is an initiator. There's a Tata box here. This is usually the promoter region. There's a recognition element here and here that are the boundaries of where the promoter are. There's a downstream promoter element, which is after the gene and affects the expression of this gene. And then there's the motif 10, where is he? Motif 10 promoter, which is after the initiator and regulates the efficiency of transcription. I am never going to ask you to draw all this out. I would like you to take this home in your brain and be like, holy cow, how can all of these things be regulating me? And then ask, why do I need to regulate my genes in such a complicated way? Because when we look at your eukaryotic genes, one of the things I need to regulate is, do I need it in this cell right now? Do I have access to the promoter? And then maybe I want to wait until you hit puberty to access it. Or maybe a specific environmental control is going to allow me to make this protein, but then I might want to do it really, really fast. So there are a series of steps that your body can go through to basically pre-prep all of your available in that cell transcript code. where you're going to actually make the transcript from the promoter on down. And you can regulate how many, when, and at what levels of hormones or at what environmental stimulus those molecules are going to bind to parts of the DNA and increase the transcription or start the transcription rate. So basically you can be ready, but you don't have that one last piece. And I always imagine the eukaryotic cell transcription process being very much like the old cartoons or movies where the guy is going to puts dynamite in the cave, and then he runs a pile of black powder all the way back. It's ready to go in many cases at any point in your life. What you need is a spark, and that spark then provides the impetus for the actual explosion to happen. It goes down and explodes in the cave. Here, all of these things are helping to regulate it at the right time. You might have enhancers. They can either be on the side of this right with the gene or far, far away. They can be on the other side of the chromosome. They can be, in some cases, right next to the chromosome. This can maximize transcription, like make the maximum amount. They are often binding sites on the DNA where the thing that is triggering transcription, the initiator, is actually being bound to the DNA. Once those enhancer regions are full, then transcription is going to be at its maximum amount. We have insulator edges here that I have my insulators, the BREs, that prevent the enhancers from overreaching themselves. And then there's silencers, molecules that bind to the DNA and prevent transcription or repress it or slow it down. Transcription factors are all the things, and there are many of them, that regulate transcription. Often they target sites on the same chromosomes, the cis-acting sites that help regulate expression or transcription of those genes. We have activators and repressors. These are different words than we used in bacteria, but very similar concepts. Activators increase transcription. So we're not having a cyclical AMP bind to camp model here. We're having molecules that that signal the needed increase in transcription, and repressors that slow it down. These are not the same thing as binding to the operator. In this case, there is no operator. These are often sites far upstream or right next to the actual code for the transcript we're transcribing. What it means is that we can get really, really good at producing exactly the right amount of transcript, so we don't waste any energy by making extra, because that's wasteful, or make it when it's not appropriate. So I want to talk a little bit, like really briefly, just give you some highlights, because I think that this is not particularly accessible to us as something to talk about beyond this. You know what I mean? It's like, this is a lot, and they're all little pieces of things. So I want to give you a general overview of what is happening here. Here is the DNA. Here is the Tata box. There is the promoter. When we want to activate transcription, we have a series of general transcription factors. That's why I didn't make this red. I just want to talk about it. The transcription factors are activated and repressed by causing changes to RNA polymerase II during the actual process of transcription. The DNA loops around and bends in on itself, and there's actually a protein called a DNA bending protein that can bring the region on the DNA that has the enhancers or the silencers or the repressors bound to it. It can be somewhere else entirely, but it is a cis-acting area. It folds it and puts it in proximity to RNA polymerase. When the DNA is folded and enhancers, for example, are tied to it, that DNA enhancers comes in contact with RNA polymerase, and that's like the spark that ignites the burning chain of black powder that actually sets RNA polymerase to go. Here there's a DNA bending protein right here in this model. This is RNA polymerase. These are all of the little factors that are facilitating transcription. These are enhancer molecules that are actually attached right here. As the enhancer molecules come into contact with the RNA polymerase, there are a series of co-activators, other things, proteins or molecules that are facilitating transcription. They have to be there. They form this really complicated, right here, enhancosome. I feel like they're just making words up now. I guess they always were. The enhancosome interacts with the transcription complex, or the proteins that are actually going to zip off and do transcription. If these are repressor molecules, it's going to prevent that from happening. If these are enhancer molecules, it's going to facilitate this happening more frequently and more effectively. And so that promoter complex and RNA polymerase down the DNA and transcribe. A little highlight about one of the most well understood eukaryotic transcription mechanisms, which is the GAL gene system in yeast. There are 4 structural genes and there are 3 regulatory genes. These genes are only needed if the cell is exposed to the sugar galactose. This is a positive regulation, positive control. The activator protein has to be present in order to turn on gene transcription. It is inducible. It is regulated by the presence or absence of galactose. This means, kind of like the E. coli lac-operon or the trip-operon, we only want to use it when it's energetically favorable if you're a yeast. So if there is no galactose, you don't want to transcribe. Now, it's not an operon. I can't just turn it on or off. So I have all of this DNA region regulating what I'm going to do or not. So I want to give you some quick highlights for the GAL gene system in yeast. It is regulated by a protein called GAL4, which is encoded by the GAL4 gene. So short version, the yeast makes a protein that represses, it's negatively regulating the genes involved in this galactose metabolism. The DNA has a region that binds either the regulating protein for this activator or GAL80 protein, which I'll have in a second. So many GALs. The genes that are actually involved in galactose metabolism that the yeast needs to break down galactose are 1 and 10. Both of these genes are positively regulated. They're controlled by a central control region, which is either activated or deactivated. It's an upstream sequence from this gene. It has 4 sites that bind to GAL4. So the GAL4 that is regulated negatively by the cell is a gene that makes GAL4 proteins. Those GAL proteins, those 4 fours travel to a part of the DNA where they bind to areas and activate the gene. The DNA folds over and then binds to the region that starts transcription of GAL1 and GAL10, which are involved in galactose metabolism. The region here, this UAS region, is basically adding enhancers to facilitate increased and more efficient transcription. I had to think about this a lot, and I still am having, you can hear, I'm having trouble explaining it. It's a lot of steps, but the overall function is cell makes many of these. The more of these it makes, the more likely it is going to enhance or turn on, bind to all this region of the DNA. These little binding regions are signaling that there is a great need for galactose metabolism. They will facilitate transcription of the genes that actually break down and metabolize galactose, the sugar. Finally, because I know you're like, wow, And I don't want to do too much of eukaryotic transcription, honestly, because I think that we are still learning a lot about it. We have some basic general ideas, but when I went to go look at papers to try to get you some better images and things, a lot of it is using that model to model how other eukaryotes might be doing it. I think we have a pretty solid idea of the yeast system for one particular gene, but eukaryotes are really complicated, and sometimes multiple chromosomes are involved in regulation. So what we want to do is start developing a database and trying to understand how they work. That's the role of the ENCODE database, and I've linked it here. I'm going to try to get some time where we can look at things on the computer and look at, because a lot of the modern bioinformatics are just things we are going to have to look at on the computer and we can analyze. The goal of this program is to identify all DNA sequences that are actually functional and what turns them on, what turns them off, what enhances them, what silences them. How do we regulate them? So what this database has been able to identify is a substantially huge amount of our genome is either regulatory or leftover artifacts from evolution. We used to consider this all junk DNA, but it turns out most of our DNA is actually regulating the little bits that actually encode for what we need. 90% of the things that cause diseases are variations located on regulatory regions, which means it's not necessarily the gene that is malfunctioning, so we think. It may be the regulation of turning it on or off, or how efficient we are with it, and that's what this database has been able to help us start identifying. It is looking for enhancer and promoter elements. Promoter elements, regions of the DNA that are facilitating or starting RNA transcription, are found throughout your entire genome. We've identified 30,000 of them. We also have started to identify a substantial number of enhancer regions. Hints for studying this: these words mean the same thing in English that they mean in genetics. Enhancing means makes it a little more. Promoter means let's get it started. Enhancer RNA enhances the sequences on promoter elements. It may be coded for by a piece of the genome that comes from somewhere else. It could be outward from, this is kilobases or thousands of bases. These are pieces of RNA that are made entirely to then return back to the DNA and enhance transcription. We don't know that much about them. This is, I guess, again, it's a new science and we are learning a lot. I feel like for these last couple lectures, I'll cut them shorter because a lot of this stuff is theoretical at this point, or we just have a little bit of pieces and I feel like in some cases, probably telling you nonsense. In a decade, you'll be like, what were we even learning? But this is how we understand it now. We know that these exist. We know they regulate transcription, but we're not sure how or why. With that fabulous ending to the lecture. Nobody knows what anybody's talking about, but we know that all, except for 5. After 5, we're pretty sure we know what we're talking about. Review this. Consider this in context of the other stuff we have covered. As we move into the final exam level stuff, it really is asking you to think above and beyond probably what you've studied in genetics so far. We've done, I know, some new concepts for you and as we've gone through the course, but now I think almost all of this is something you have not been given in other courses. So be patient, ask a lot of questions, but also recognize that the science here, these databases and papers that I was pulling to try to explain this to you a little bit better were being published in 2023. So really new stuff, and I'll try to give you at least a basis to ask questions about it when you take this into your research or in your careers later, so that you have some recognition of this. With that, that's it for the whole week. I will see you in lab. Hopefully everything is working, and I will see you in the exam this week. All right. Bye-bye.